So welcome to Oracles tonight, and we choose tonight a little bit of a different topic, moving towards glaucoma and glaucoma diagnostics. And we just had a brief introduction discussion here among the speakers about the idea of measuring intraocular pressure over the whole day or over 24 hours, 7 days a week. Over a longer time period has evolved decades ago, and with the involvement of technology, especially of the last 15 years, we have seen multiple approaches and attempts to use more 24/7 intraocular pressure measurements. And I think today what we would like to do is first of all have various presenters talking about innovative technology that is in the market or is coming potentially to the market. And then I think we will open up the floor for an exciting discussion about where's the technology today, where it might come, what are the roadmaps, and what is the exciting elements of measuring intraocular pressure throughout the day, throughout a continuous period of time. With us, we have well-known speakers, Kawek Mansouri from Switzerland. We have Zeynep Aktas from Ankara, Turkey, and Aylin Crunkleton from the US. Sorry about that. So and Noam Hadas from Israel. So it's a great pleasure and a great joy to have these guest speakers here. And Kawek, it would be great if maybe you can give us a first introduction presentation about the topic.
Thank you, Michael, for the kind introduction, and thank you to the organizers for the invitation to speak here. It's a pleasure to bring a glaucoma topic to colleagues from other subspecialties. I will start with my PowerPoint presentation. That's the more formal part, and I'm sure we'll have ample time for discussions afterwards. So I'll give you a brief overview about 24-hour monitoring and devices that I've been involved with myself, and these are some of my financial disclosures. As you all know, of course, intraocular pressure is the only treatable risk factor for glaucoma, but it is a highly dynamic parameter, biological parameter, and our gold standard GAT, Goldman Applanation Tonometry, provides only a snapshot of IOP behavior. Your IOP will change from minute to minute, hour to hour, based on a host of extrinsic or intrinsic parameters. And here you see a list, a non-exhaustive list of events, activities, foods that may change your IOP. Even this presentation may change your IOP, or hopefully positively. That's why getting beyond measuring IOP simply for a few seconds using GAT or eye care or other devices, having continuous measurements has really been a quest of colleagues in the glaucoma field and beyond for at least 2 decades, actively for 2 decades. So the first approaches were for 24-hour monitoring, just one 24-hour period, and they would go by using extraocular sensors, typically integrated in a contact lens. The first such contact lens, the first smart contact lens actually ever developed, was developed in my city, Lausanne, by 2 young PhD students who I had the fortune to meet almost 20 years ago when I was a resident myself. So we developed what then became the triggerfish contact lens. This triggerfish contact lens is a soft silicone contact lens, and it has strain gauges towards the periphery that will be located around the limbus when you put the contact lens on the eye. And they measure changes of the ocular circumference continuously or near continuously. So initially, we were under the assumption that those measurements would reflect IOP changes alone. As we did more studies and then clinical trials, we understood that the measurements do not only reflect IOP changes, but also reflect intraocular volume changes. And the two are not always the same. They don't behave in the same manner. And then the third parameter, which it measures or which affects the measurement strongly, is the ocular biomechanical properties, or how elastic or rigid the eye is. The other problem we faced was, although the measurements seemed to be quite accurate and relatively reproducible for such a biological dynamic parameter, but the other problem was that those measurements, we did not manage to translate them into millimeter mercury values, which means that once you get your 24-hour measurement, as you can see here, the patient comes back to the clinic, you take off the contact lens, you download the data, and then you see how the measurements behaved over 24 hours on this graphic. You see their fluctuations, you see the timing of the peak pressures, typically at nighttime, as you can see here in the middle, in the grayscale, when the patient is asleep. You can see all of that. But on the y-axis, you do not see those measurements reflected in our millimeter mercury scale. You see them in millivolt equivalents. And you don't really know what that means in reality. So that was the big challenge we faced and couldn't solve, unfortunately, till today, which makes the clinical use of it very difficult or somehow difficult. Because again, you don't know what the absolute measurements are, but you do see how stable or unstable your IOP is. You do see when the peak values occur. And this is the reason the device got FDA approval in the US. And in Europe, it was approved before anyway. But it got an FDA approval for a very limited type of monitoring to find the time of peak IOP, which then would need to be confirmed by standard tonometry. But what happens if the timing, as so often, is at nighttime and you need to confirm it by tonometry? You can't really, because, well, the patient is asleep and the ophthalmologist is asleep. So the commercial use of this device really didn't happen because of this difficulty. So another way to measure IOP, and measure it now in absolute values, where you get millimeter mercury equivalents, or millimeter mercury values rather, is by implanting a sensor into the eye. And the first such approach was done by a German company called Implant Data. The device is called iMate. It was approved in Europe. It was CE marked 5 or 6 years ago. And there are two types of the sensor. The first type is a sulcus-based sensor, which you can see here. The idea is to implant it during cataract surgery in front of your IOL, inside the eye. And this device is intelligent in the sense that it has, an ASIC, and pressure sensors, and so on. It's like a small computer that you implant in the eye, and it measures absolute IOP inside the eye. And the data are then transferred to an outside reader, which the patient brings, like an iPhone, close to the eye. And by doing so, he obtains a measurement. You can see his measurements, and this device sends power to the sensor. The sensor doesn't contain a battery. It is wirelessly powered each time the patient obtains a measurement. If a patient wants to have nighttime measurements while he's asleep, he can put a sleep mask on and it's done automatically. So that's also possible. The placement, again, is in the sulcus. And we saw in the first generation that there is some degree of pigment liberation, especially if there was manipulation during the surgery, which fortunately did not become progressive. Otherwise, the device was quite safe, but it was limited to cataract surgery patients. Now, you have a glaucoma patient who doesn't need cataract surgery. What do you do? Well, we developed another sensor, same approach, but different shape. We call it the iMate SC, supracorridor, which you can see here. It's rectangular. It has a 3.5 to 7.5 millimeter dimension, and it's implanted during glaucoma surgery, such as deep sclerectomy here or viscocanalostomy, instead of the regular passive space maintainer implants that we use. We usually use a collagen implant or a snopper implant or PMMA implants to keep that surgically created space open. But these implants are a bit silly, if you like. They are passive. They don't do anything. They just sit there to keep the space open. So we thought, let's make those space maintainers smart and implant a sensor instead of a passive implant. So here we are doing a regular deep sclerectomy. Here I'm taking away gently the deep scleral flap, creating the so-called trabecular Descemet's window through which aqueous percolates outside of the eye without penetrating into the anterior chamber. That's the difference to trabeculectomy. Now, that's the new step. Here, I create a small incision to go inside the suprachoroidal space, which I opened up by putting some viscoelastic device to create a pocket. And here is a sensor. This is the part that has to touch the choroid where it measures. And this was the very first case I did. And despite it being the first case, you see it's not particularly difficult. So here I'm putting it gently, just pushing it in a bit. Half of it will stay as a space maintainer, and the other half will go into suprachoroidal space. And then you close the flap, close the conch, and voila, your surgery is done. This is the same eye the next day, nice filtration bleb, no sidel, eye is quiet, anterior chamber is deep. Here on anterior segment OCT with the anterior, you see the space that you created, the implant which is there, it occupies a large space. And then the patient is there, happy, and he goes home and he starts measuring his pressure whenever he wants. These are the little pink dots, and you see right away how his pressure behaves without the patient having to come to see you. Well, this was investigated in a multi-center study, 4 centers in Germany and ours in Switzerland, in combination with non-penetrating glaucoma surgery. We found that the measurements were very reliable when you compare them to GAD, our gold standard. The correlation was around 0.7 to almost 0.9 according to the statistical analysis you used. Now, it's not 100%, but you may argue that this device, which is really inside the eye, may be more accurate than Goldman tonometry measurements that measure transcorneal pressure difference. We have published recently first the 6-month data and just last week the 1-year data in ophthalmology, where we demonstrated that there were no serious adverse events. There was only 1 case of corneal detachment, which we solved under medical management. Again, not unusual, out of 24 non-penetrating glaucoma surgery cases, and everything else was similar to what we see regularly. Another paper which I'm citing here, but I think was important and was recently discussed in detail in Glaucoma Today trade journal, was that during the first lockdown period of COVID, if you remember it in May 2020, when we couldn't see our patients, we had the fortune in those patients who did have the sensor in the eye to be able to monitor them remotely to see what happened to the eye pressure. Here's an example of how unstable eye pressure in a well-controlled glaucoma patient like this lady can be. And these are measurements over 950 days. You can choose any given day on this graphic, and you see that the pressure can range from as low as 5 to as high as 20 or 25. So this is how unstable a well-controlled glaucoma IOP can be. Now, we have looked at the data that we have to study different questions. We have a lot of data. Here you see in this publication, we had 22 eyes included, but we had over 100,000 IOP measurements from almost 16,000 measurement days. So by far the biggest IOP collection ever. And we could study questions like, for example, if there are seasonal variations, but which they are. We found that statistically significantly IOP is lower during June or July compared to winter months. So in summary, your IOP seems to be doing better. And it's not a small difference. It's an average difference of more than 1 millimeter mercury. Imagine that on an individual level that can be higher. And imagine how important this difference can be for pharmaceutical trials of one medication compared to the other. I'm going to finish here, but I think based on the experience that these devices and others have given us and will give us in the future, we will now become able to offer better routine IOP monitoring, hopefully routine remote IOP monitoring as well. This, I'm sure, will improve glaucoma management. This will better enable us to use sustained release IOP treatments, because if we are not going to see patients that often because they have a sustained release for 1 to 3 eyes, at least we should be able to monitor them remotely. When patients can check their IOP, I'm absolutely certain that they will have better adherence to their glaucoma medications. Their quality of life will improve because they are less scared of missing IOP peaks. They can monitor the IOP like diabetic patients can. There are challenges, however. We still need to find out about long-term safety, but I can tell you that there is one patient at least who has had the eye-made implant in his eye for 10 years. So there are more patients getting to the 10-year point now, and we can see that the device is still safe and still functioning. We will need probably to better use AI to interpret this huge amount of data and maybe even reevaluate how we practice glaucoma when we have much more IOP measurements. Thank you very much.
Thank you. We would like to have the discussion at the end of all the presentations. So what I would like to ask Noam, if you could share with us your technology.
Good evening or morning or wherever this applies to. I'll be happy to share the technology. I just want to warn everybody that I'm not a physician, and I understand very little about eyes. I come from the technical side of things. I'm an electronics engineer, so be careful about the biology or anatomy questions. The device we are developing is called the, just a minute. Yes, we call the device the IO Perfect, and unfortunately, this is not a continuous 24 hours measuring device, but rather it's a point measuring device, but the measuring process is so simple and so quick that the patient can repeat the measurements as many times as he wants during the day. And if really necessary, he can wake up during the night and complete the measurement in less than 2 minutes. Just so that you'll understand what the device looks and works like, here's a short video. Now, the measurement concept is totally different than, as far as I know, anything that ever been done. Basically, what we do is use these goggles to apply an external, a constant air pressure over the eye. And video cameras look at the response of blood vessels, mainly arteries, in the eye, over the eye, to these changes of air pressure over the eye. And signal analysis of these responses allows us to calculate the intraocular pressure. If you look at the general description of the eye, you can see that above the eye is exposed to atmospheric pressure. Inside the eye, you usually say that the pressure is 15 or 10 millimeters mercury, but it's not. The absolute pressure is the atmospheric pressure plus the IOP. So if the atmospheric pressure is 760 millimeters, inside it will be 775 millimeters mercury. And you should remember that. When you look at overall the vessels in the eye, you can see that they are generally divided into two parts. There are the vessels that go over the eye, over the sclera usually, and these vessels are only exposed to the ambient pressure. But after traveling some distance over the sclera, many of these arteries, mainly the anterior ciliary artery, they dive right in and continue their journey inside the eye, where they are exposed not to the ambient pressure, but to the ambient pressure plus the IOP. I take this fact and use what is known as the oscillometric method for blood pressure measurement. If I look at the external arteries, the arteries that go over the eye and are exposed only to the ambient pressure, and I increase the ambient pressure from 0 to some level, what you will see is that the vessels start to pulsate when the ambient pressure is between the diastolic and the systolic blood pressure inside these vessels. So what you'll see is that they initially don't pulsate very much, then the pulsation amplitude increases to some maximum, and then the pulsation drops again when the external, the ambient pressure is higher than the systolic pressure, so the vessels collapse totally. The same thing happens to the vessels inside the eye, but they sense a pressure that is IOP higher than the vessels, the external vessels. So you'll get the same thing, but the whole response will be shifted by some level. If I compare these two graphs of amplitude versus pressure, you will see that the difference between the peak at the internal pressure and the external pressure is exactly the IOP. And this is a direct measurement of the IOP using pressure. This is, for example, one of such measurement. These vessels are inside the eye. We find them on the iris. These vessels are outside of the eye. We find them on the sclera. We use video cameras that look at the eye while the air pressure above the eye is changing. We do a rather sophisticated signal processing, image processing, and signal extraction algorithms to arrive at these waveforms, at these peaks, and get a measurement. The first question which was asked was how painful this whole process is. So we did do a small clinical study where we used the device on quite a few patients, and luckily almost everybody said it was perfectly acceptable because the pressure was not too bad, not painful, and so forth. We are now struggling with video and signal processing. I'm optimistic, but we don't have a product yet. Thank you.
Thank you very much. Great, interesting pathway of going towards the IOP to a different approach. Let's hear what Aylin can share with us.
Okay. So, well, first of all, I really appreciate being here. Delighted to be here. This is a great panel and a great opportunity to introduce you to iOptic. And I'll start with a little bit of background on the company, which is located in Lexington, Kentucky, here in the U.S. And I recently joined the company as CEO to help with commercialization. But the company was actually started some years ago by Dr. Britt Brockman, who's an ophthalmologist who, like many ophthalmologists, as I'm hearing here, he wanted to find a simple way for patients to measure their IOP. And he was on the board of trustees at the University of Kentucky, where he was introduced to Professor Todd Hastings, who is a professor of electrical engineering with a focus on nanoscale science. So they teamed up, starting with a clean slate to develop the product. And one of the things that always comes up there, early research told them that to get the accuracy and repeatability they thought needed for the long term, that the sensor would need to be implanted. So it would be in direct contact with the intraocular fluid. So they came up with concepts, they spun the technology out of the university, and then under small business grants from NIH, they developed the core technologies that form the basis for the product that we're commercializing. And I'll talk about that a little bit. So our solution is a device that has a pressure sensor that's permanently implanted and a handheld reader. The pressure sensor is a mechanical diaphragm, which is a very thin membrane that changes shape as the pressure changes. The handheld reader has a light source that illuminates this diaphragm and a camera that takes a picture of an interference pattern that reflects back, which gives us the pressure to within 1 millimeter of mercury. This sensor is then mounted inside of an anchor, what we call the anchor, that gets attached to the iris. And I get a lot of questions about durability and what have you. What we did is we borrowed the experience with IOLs, particularly anterior chamber IOLs, to use designs that they used, the materials, that kind of thing, and to help to avoid problems like endothelial cell loss, for example. And what we do is, if you look at the sort of the envelope for an anterior chamber IOL, our implant, the complete implant, fits within that envelope. So our envelope, the implant fits within the envelope and made of the same materials. So I think you can appreciate the simplicity of the design. The the implant is just this mechanical diaphragm with no implanted electronics. We expect this to last for the life of the patient. And we make these sensors using a MEMS manufacturing process. So they're made by the thousands, and they're pretty inexpensive to make. And here's the complete product, which has the pressure sensor implant, an insertion device, and a handheld reader. The pressure sensor is implanted during a combination procedure, so either cataract or glaucoma surgery. So, for example, once the cataract surgery is complete, the pressure sensor is inserted through the same incision that was used for the cataract surgery, and attaching the implant to the iris is complete in less than a minute. The handheld reader, the patient that holds the handheld reader to the eye and presses a button to take a picture, and it literally just takes a few seconds. So then the data then is collected and stored in this handheld reader, which is then shared with the doctor's office using a cloud-based connection. So it is similar to the plot you saw earlier, except this is hypothetical. Once a month, the clinician reviews the data. Here's an example where the clinician watched the IOP increase, the average IOP. Obviously, there's data all over the place. Take the average IOP each day. And once the average IOP reached 18 millimeters of mercury, an eye drop is added. Then the pressure settles at around 15, except for a couple of times when it looks like the patient skipped a drop or two. And that's important, because not only will we be able to monitor whether the treatment is working, but also whether the patient is complying. So that's our product. And you say, well, how does it compare to all these other products that are, I just quickly put this together last night, a list of companies that are currently active, and I'm sure there are those that I've missed. But we believe that the reason that there's no product that's getting mainstream use now for glaucoma patients is because no companies had the technologies to offer the complete set of requirements for success. In particular, accuracy of 1 millimeter of mercury, and with this mechanical diaphragm, there will be negligible drift. Ease of insertion of the implant, less than a minute in a combination procedure. Ease of patient use. There's no contact lenses or disposable probes for the patient to deal with, and taking the measurement will take only a few seconds. So basically, we think the patients will use this device. Durability, we've talked about for the life of the patient. And as far as affordability, we expect our clinical trials to show high value superiority over the current practice. And combining that with a low cost of goods, we expect reimbursement to be reasonable for the payers and attractive to our investors. And with that, I'll just end by saying that we're, at this point, we've tested all of the core technologies using benchtop, short-term, long-term animals. Everything's been thoroughly tested. And right now we're raising a round of financing to do first in human cases. And once that's done, we'll raise another round of financing, and we will be planning to launch the product both in Europe and in the US. And that's it.
Oh, thank you very much. I think I would like to open up the discussion with, and I'm very happy Zeynep joined us, and I recognize Zeynep is a very practical clinician. So Zeynep, you saw now multiple technologies, starting with the triggerfish. We have implant data. You have a device that can be used at home, more like 3D goggles. And we have seen another pathway of implant technology. So very practically, is that for all patients? Do we have to select? Is it some for children, some for adults? How would you choose which technology to use for which patients?
Thanks. First of all, thanks for the invite. It's so exciting to see these developments and what's happening around. So I really believe that future is there. So this is going to be the future. So we will be using so many technologies in the future in terms of IOP measurements. I really believe that, because our aim is not just simply to reduce intraocular pressure. We know that. We need to know the IOP pattern. So we need to modulate. So we need to know that IOP is stable in our patient, because it fluctuates, it's dynamic. So we need to know what's going on out of the office. So that's for sure. I believe we need to select some patients, of course. In my clinical practice, if I can use these technologies, I would really like to use these technologies, for example, in kids, that's for sure, because we cannot measure intraocular pressure in these kids. Maybe we'll be able to do this in the future. Another patient group might be the best fit. For example, there might be the normal tension glaucoma, which is a debate. What I really need to know what's going on out of the office. For example, in some patients you can see the IOP is stable, but the disease is still progressing, so you don't know what's going on. So in these patients, these technologies would be very useful. And on the other hand, from the practical clinical practice, we need to develop individualized treatment patterns in the mixed era. So these technologies would definitely give us a chance to individualize these glaucoma treatments. So I definitely agree with Kawek. So these technologies is going to increase patients' adherence. So this might help in turn us to individualize our glaucoma treatment. So I definitely believe in these technologies, but I wish I had a chance to use this technology in Turkey. The problem is cost here, So yes, we should select some, especially some patients, but because of the cost, I don't know, is it possible to use all of these technologies in every patient? So definitely we should choose.
Okay. Kawek, maybe a question to you a little bit on the line is you worked with triggerfish, you worked with implant data. So how many of your patients, what percentage gets those technologies today in your practice?
Well, not many, and there are two main reasons for that. With the triggerfish, the device is costly and it's only for one eye, one time. And then you need to pay again the same amount of money, which limits it. And it's not routinely reimbursed by insurances, even here in Switzerland. We have to ask for reimbursement, justify it, and then we can go ahead. So that already limits us to a small subset of patients. These are typically patients who are progressing despite IOPs that are low. And I should say these are office IOPs that are low. So we see that despite that they progress, and we want to find out why that is, and if there are peaks, when do they occur, and then decide what would be the best way to address those peaks. Oftentimes, we move to surgery anyway, because we see, again, there's always some sign of progression. And once we do a 24-hour contact lens monitoring, we see that the pressure is usually highest at nighttime, where most medications, drops don't work. And the ultimate attitude would be surgery. For the patient, now having seen a 24-hour monitoring visually, where they see IOP at daytime is maybe here, but at nighttime it's here, although we can't tell them how much that is, but visualizing IOP behavior makes it easier for them to accept surgery, to undergo surgery. Again, these are patients who have no low pressure. For example, office IOP is 12, 13, 14, and they're progressing. So it's hard to sell surgery to these type of patients. But if they see the fluctuations and the nighttime peaks, that can help. So that's for the triggerfish. Just one patient category, the typical one where we use it. The other one is with the iMate, the implant data iMate, the intraocular sensor, which is also CE marked. So we can use it in Europe commercially outside a study setting. Here, the cost is not an issue. I think it's quite economical, especially because it stays in the patient's eye for a lifetime. The problem we face now is manufacturing of the sensor by the company. So we have ordered more than the company can deliver. So here there is a shortage of the device to be sold on the market. And then the other limitation, of course, would be we could not offer it to everybody, like the contact lens, but only to patients who need to undergo glaucoma filtering surgery.
I want to make very quick comment. Maybe Suphi may join me or not. I don't know. As a refractive surgeon, we need to measure intraocular pressure without independent from the cornea. I think this would be amazing for us because we never trust, of course, normal measurement from the cornea as we change corneal shape and thickness. I just wanted to make a comment importance of refractive surgery. Maybe Suphi, any question or any comment?
Thank you, Aylin. I absolutely agree with you. And thank you to all the speakers for these enlightening presentations. I learned a lot, and obviously the benefits of 24-hour monitoring are very clear to even a lay person. However, yes, I would love to have a tool to have 24-hour continuous measurement, which is accurate, but at the same time, not invasive. And I think there's the contradiction. The more we go inside the eye, the more the accuracy increases, probably. And the more we are outside the eye, the less invasive, the less risks we have. So there's this spectrum that Kawek described, from triggerfish to the intraocular devices that are put into the sulcus. It's hard to justify making an intraocular surgery just to make some measurements before you then do some glaucoma surgery. So yes, I agree. It would be nice to have intraocular solutions that can be done at the time of cataract surgery or glaucoma surgery, like the ones we have described by Aylin and also by Kawek. But it would also be nice to have a more precise, more accurate 24-hour measurement, maybe inside the choroidea or outside, like a scleral explant that is sutured to the sclera. Do you think this is coming? Do you think, Kawek, your device could be miniaturized so that it could be injected into the sclera or sutured onto the sclera, maybe it would lose some accuracy, but it would be less invasive. And I love the new concept to me of having a device put on the iris together with the cataract surgery as well. But as you said, not everybody needs cataract surgery, and it would be hard to justify such a surgery just to prepare the patient for yet another surgery.
Suphi, thank you for these very valid points. I will try to give you a short answer so that there is more time for other questions and other panelists to answer. The question of miniaturization is very important, and it is coming. The current generation of the iMate was developed about 5 years ago, almost 5 years ago, and then you need to go into clinical trials and then have the follow-up. And only recently, the 1 to 2 year data became available. In the meantime, of course, the company has been working on miniaturizing it. And this is happening. It will become smaller. For it to become injectable, which means not needing intraocular surgery, just a simple injection, there is still a bit of path to go. So that won't be available immediately in the coming years for sure, as the earlier the better. And then there would be different places where it could be injected onto. So you mentioned that it could be injected either into the anterior chamber, somewhere where it's stable, it doesn't fly and float around and damages the cornea, maybe somewhere in the trabecular meshwork. It needs, again, to be stable there. We don't want it to touch the endothelium to damage it, or it could be somewhere in the posterior chamber. It could be somewhere in the choroid, or the junction between the sclera and the choroid, which is a place I personally favor, because this is really where posterior chamber IOP can be accurately measured. And the one IOP that damages the optic nerve is the one closest to it, meaning in the in the posterior and not in the anterior chamber. And also, injection-wise, it is a place that can be relatively easily reached. So yes, absolutely it is coming, and technology is advancing faster than regulatory approvals happen. So that's one limiting factor. Every 3-4 years, technology allows us to miniaturize by a factor of 2 or 3 or more. But by the time we get approval to conduct studies, then the necessary follow-up, then the submission to regulatory agencies, and then answers. This takes many years. The clinical validation is where failures happen and where success can happen. But the reason so many of the IOP monitoring pilots that I've seen myself over the last 15 years, either as a reviewer for grants or as a reviewer for publications, and there were many of them, very smart ideas, but only 2, the 2 I mentioned, have been approved for clinical use in Europe or in the US. Only 2 managed to go through clinical validation and then be accepted. That's where the huge difficulty is, not really the technical part.
Thank you.
Zeynep, do you have a question?
If I have time, yes. So I would like to make a real quick comment on the corneal biomechanics. So as you reminded, so is it that is a little bit conflict? So we are using technology. So we try to get rid of corneal biomechanics and IOP measurement errors, But on the other hand, we try to use contact lens, Sensimed, relying on corneal biomechanics. Right. So what do you think? Could you please make a comment on? So how would you choose your patients according to biomechanical properties of your patients?
That's a good comment, Zeynep, and I don't have a good answer to that. So you're absolutely right. The measurements of the triggerfish depend on the cornea and scleral biomechanical properties. To which degree those influence the reading, we couldn't find out. We haven't found out. So I can't tell you if it's a more rigid, for example, you mentioned corneal hysteresis and elasticity, or if the thinner cornea. I can't tell you if it's thinner, the signal is biased by that much, or if it's thicker. We don't know that. That's why I think the best approaches are intraocular.
Yeah, exactly.
Again, the main advantage of the contact lens is it's non-invasive, so everybody can benefit. Some patients will be adamant they don't want any type of surgery just for diagnostic purposes. So
It just gives you an idea, you mean, right?
Exactly.
Okay.
And the other limitation is only 24 hours. And we have published that, you know, short-term IOP is not very reliable. Short-term meaning for the duration of 3 months. So if you measure today, it gives me an R value of 0.5 for what will happen in the next 3 months. So 50% is explained by today's behavior. But for what happens beyond 3 months, this value goes down to 3 to 4. So only 3 to 40% of what happens beyond 3 months is explained by a single 24-hour monitoring. So really not good. Of course, it's still much better than one single GAD measurement. It's very low.
Thanks so much for your comment.
No, I'm actually along that line. A question to you. I mean, you choose a technology where you modify the environmental pressure and to see if the pulse amplitude in the arteries or the veins are changing. Do you see any influence there? If you change the outer environment pressure, do you see any influence on the intraocular pressure? And there was actually a question from the audience. There seems to be publications that there is a influence on the IOP if you modify the environmental air pressure.
Yes. Actually, there are publications that demonstrate that continuous pressure on the eye will eventually lower the IOP when the pressure is removed. I guess this is because higher pressure on the eye increases the pressure inside the eye and leads to more perfusion of liquid outside. But this process takes time. And the whole process of pressure application and release takes 90 seconds. So we don't expect significant changes to the IOP during the process. By the way, we did confirm that by taking a Goldman measurement before and after our measurement on multiple patients, and the IOP did not change at all.
Maybe along that line, so where the other technologies presented are mainly implants. How do you see your device being used by the patients? Is it at home? Will it be in the office, at some point of care? Where is this device going to be used mainly? Where do you envision that?
Well, I guess that's mostly an economic question more than a medical question. Naturally, some patients will need the device at home because once confirmed that the glaucoma is highly variable, that the pressure is highly variable, the physician would like to know long-term what's happening. But even before that, I very much see the device in pharmacies, in places like Walmart or Walgreens, I'm sorry, where a walk-in can come in and get a blood pressure measurement and get an IOP measurement using a device which is much more convenient and definitely more accurate than the pulse tonometer that they are using today. Even when waiting in line to get your eyes tested, the physician can save a few minutes by having the nurse measure the IOP before the patient comes in. So the economics will decide where the device will be used. It's not going to be very high cost. The device will be a few $100s and no cost per measurement. So overall, it's rather straightforward.
I have one question for Noam, if I may.
Of course, that's why we're here.
So thank you, Noam. I'm very interesting concept, and congratulations to you and to your team for that. So I'm very interested, and I have two brief questions. How are you thinking of validating the device? Because obviously, when you're wearing the goggle, you cannot measure by tonometry. So how can you validate it? That's my first question. And the second, do you have any idea of how changes to blood vessels that happen in different pathologies over age may affect also the measurements and the readings you obtain?
Thank you. The first question is quite simple. As I said, we do a standard tonometry measurement right before and right after each one of our measurements. And if my measurement is in between, I believe we can show that we are at least as accurate as the gut. Maybe in the future, we'll use one of those implanted tonometers to get an even more accurate measurement, because just inside this forum where I feel free to express my engineering opinion, the Goldman is not accurate. Okay, if it was me, I would, go to 3 different physicians and get three different results. Not greatly different, but often different. That's one. The other question is, I look at the whole eye as a mechanical device. Anything that happens during the measurement process is just, the very basic mechanical interaction between a tube with pulsating pressure inside and a fixed pressure on the outside. The properties of the tube are totally irrelevant. So I don't see how the properties of the vessels, of course, if there are no vessels, I will have nothing to analyze, but then there will be no eye or the patient. I don't think any vessel disease or eye condition would change the accuracy or the feasibility of this measurement.
Michael, if I may.
Yeah, sure.
Noam, I think you are absolutely right. Gut is very flawed, and Goldman himself knew this when he invented it. And so one of the biggest mysteries in ophthalmology to me is why we still are using gut at all after decades. So we agree on that. I have a question to all the experts. Are we not looking at one side of the equation only? I mean, 24-hour IOP measurement is great, and it's needed, and it's very obviously having benefits that we are missing today. But don't we need to look at the systemic systemic blood pressure at the same time?
I was going to ask same question. That's right. Especially during the during nocturnal peaks. So it's very important to see what is diastolic and systolic blood pressure in terms of perfusion, optic nerve perfusion.
The perfusion pressure matters.
Yes.
Actually, I've seen at least one paper that discusses glaucoma not as a result of intraocular pressure, but as a result of the difference between ocular and cranial pressures.
Yes.
And cranial pressure is naturally very well linked with blood pressure. So
another debate. Yes.
That's an excellent question. By the way, once we validate the device as it is now, the same device will be able to measure intracranial pressure simply by looking at vessels on the retina, rather than on the frontal, on the anterior part of the eye. So maybe in the future we'll have that measurement done at the same time using the same device.
Didn't Sensimed look at that in one of the studies between triggerfish and a relationship in blood pressure, systemic blood pressure?
Yes, that was studied, but it was an underpowered study, and it was more of an exploratory study.
Yeah.
But the thinking, of course, was there, and what my colleagues alluded to is very much true. At the end, it's plausible to assume that what matters most for glaucoma development and progression is the translaminar laminar cribrosa pressure difference. So the pressure inside the eye, IOP, and the pressure outside and around the eye. We often refer to intracranial, but we should be more precise. It's intraorbital. It's that portion of the intracranial pressure around the eye and the differential between the two. So some papers have shown that patients with glaucoma have lower intracranial pressures. And again, if you consider, especially at nighttime, when you're in the supine position, that your IOP goes up, if at the same time that counter pressure behind the eye doesn't increase to the same extent, but decreases, for example, because there's a blood pressure dip at nighttime, that then this differential becomes even bigger, and then the perfusion pressure is severely undermined. We know very little about that. Why? Noam alluded to it, because just as we cannot measure IOP well, we cannot measure non-invasively intracranial, intraorbital pressure. So there are, again, some concepts, some devices, some pilots, but to my knowledge, today, none of them has been, again, approved clinically for routine use to measure intracranial orbital pressure non-invasively. So once we have the two devices, IOP and intracranial, then we can really look at the interaction between these parameters, find out what affects glaucoma, and then the next step would be where can we attack? And then we may have more than one approach, which is today lower IOP.
You just gave me a great idea. I'm challenging the forum. How about we treat glaucoma patients by having them sleep with the head lower than their feet on a tilted bed? And this will definitely increase the intracranial pressure without affecting the IOP.
Well, there have been discussions like that, and there is a paper, for example, that looked at putting a widget pillow. I think that was a paper from Canada. So you bring up actually the head position. It's less supine. You bring up the head. You don't lower, as you mentioned, the feet, but you bring up the head, by which you would lower IOP, and so on. But many of these approaches where you lower IOP would have the counter effect of lowering, either increasing blood pressure or lowering perfusion. And in any case, we would go away from the physiological, natural behavior that we have. And I'm not a fan of changing a natural behavior that has been developed over millennia until we have good, solid clinical data. So I'm not advising my patient to change the sleep position based on these studies, because we just don't have the evidence for it.
Okay, that's why I propose a study. I need the evidence.
I have maybe a last question to Aylin and to Suphi is, we do see that more and more patients go into refractive lens exchange. So basically doing a cataract surgery in much younger ages than we did in the past, and the numbers are growing. So if you have a patient that you would see with initial potential signs of glaucoma, would you do some thought of an implant data or the technology presented by Aylin? Would you do this kind of preventive to have the ability to monitor, or is that something that's not coming? What are your thoughts?
I will answer first. My experience after refractive lens exchange, I am doing a lot. I see only in young population because of steroid. I don't see glaucoma patients after refractive lens exchange. Maybe hyperopic patients we are doing more, and maybe anatomically, of course, this is a lot of evident after lens exchange. This is very positive to glaucoma because of we are changing angle. There are so many glaucoma specialists here. I'm very afraid to tell something wrong, but after hyperopic refractive lens exchange, this is a lot of paper shows us recommending this is very positive on intraocular pressure. My experience didn't increase. I didn't see anything, but after steroid use, of course, sometimes.
Yes, I would agree. The European Glaucoma Society recommends or sees the benefits of lens exchange in order to lower the IOP. So that would be treatment of the refractive error and lowering the IOP at the same time if we did refractive lens exchange. And at at that time, it would be great to use Alan's device on the iris in a very little additional invasive procedure or step. That would be, I think, very well justified. But you could also argue in a typical cataract patients, why don't we regularly combine cataract surgery and stent implantation? I think this is something that's going to be coming as well.
Okay, I want to give everyone maybe a last final comment on the session. Maybe Aylin, you want to start? What are your thoughts? 24/7 IOP measurements to come, still under development, or are we here yet? What are your thoughts?
Was it Aylin or Alan? Which?
No, Aylin.
Alan,
You.
yes. Well, obviously, I'm hoping that we're going to see a trend in that direction. I think that you know, I'm new to this ophthalmic space, and I in the beginning I spent several months just understanding why isn't there a product already, and the conclusion was, but without the experted thoughts, is that there was just no device out there that was easy enough for the clinicians to use, easy enough for the patients to use, and gave the accuracy that was necessary. And that was the reason I was really attracted to this venture.
Thank you. Noam, what are your thoughts?
I just echo what Alan just said. I mean, even implanted devices are definitely not for everybody. Same for contact lenses. My device is cumbersome, big, doesn't provide 24-hour continuous measurement. So we need something new. Okay, let's meet in 2 years and see if we've got something.
Zeynep, is 24-hour measurement ready for prime time? Are you going to go and start tomorrow?
It's still evolving. It's still evolving. But as I told, it's going to be the future for sure.
Yeah.
So I believe, I really believe, but maybe someone invents an intraocular lens with a chip in the future. So that would be the best. Not in the sulcus, but in the back. Why not? So what do you think, Kawek? Is it possible?
Of course it is. Of course it is possible. It just takes some money and the will behind it, because money is required and patience is required to get it through all the trials and regulatory approvals.
But
so
at least we'll be able to use them in glaucoma and cataract patients. So why not?
Yes.
But we need money, definitely.
In 2010, when I started, I used to think by 2020 we would not be using GAD anymore. Well, I was very wrong. My next prediction or hope is a bit more modest. I hope that by 2030, we'll still be using GAD, but I think at least tertiary glaucoma centers will routinely offer 24 to long-term glaucoma monitoring devices to their patients. Not to everybody. GAD will still probably be used 2030, but we will have more clinically available as leading glaucoma centers.
Suphi, maybe a final comment from you is, if you wake up tomorrow morning, what are you going to change in your praxis after hearing all this?
Well, I look forward to a future where when I make the balance of risk and benefits, then I have a customized approach for every one of my patients. I would choose the least invasive solution that gets this job done. So that could be anyone from triggerfish to the intraocular implanted solution in the sulcus. I think every one solution is valid and should be available to us and our patients.
Right, Aylin, some final comments from your end?
I have I have no comments actually. I really learned a lot, and glaucoma, very special department. Also, I understood more. So I have no more comment, and my expectation from glaucoma specialist to measure after refractive surgery would be very good for us after LASIK surgery. So every year increase number of refractive surgery, and they don't know their numbers after 5-10 years later if they have glaucoma. Normally normal standard measurements may not be right, and doctors, if ophthalmologist doesn't have idea preoperative values, this might be a problem in the future. And my expectation is, of course, these devices are very good, and in my field also necessary without cornea measurements.
So from my end, and first of all, I would like to thank all the speakers and all the faculties for joining us. But for me, I really was great to see, on one hand, there is really a big gap in ophthalmology. You know, how we're going to address intraocular pressure in monitoring. I think we heard various approaches, starting from contact lenses that could be put in potentially in specific cases where, you know, the reason for glaucoma progression with low IOP is unclear. We heard the surgical solutions and implants, which get more direct measurements going direct into the eye and allow readings, maybe in combination with cataract procedures. And I think we also heard a technology that potentially could actually change the point of care, rather than doing the measurements in the clinical office with gut, which needs some trained personnel. Maybe you can do it in future in wherever, in Walmart or Walgreens or one of the other service points, maybe in optician stores, get very reliable measurements on a regular basis, where we don't have to have the expertise of doing a Goldman. So I would like to thank everyone for joining us, and hope you join us next time for Oracles. Thank you and good night.
Good night. Thank you.
Thank you.
Bye.
Bye-bye.
Thank you.
Bye.
All right.